Before we get started, I'll explain what each candy represents in the process of an action potential. The licorice represents the cell membrane, the sour circle represents the sodium-potassium pump, the blue rectangles represent the voltage-controlled sodium ion gates, and the orange rectangles represent the voltage-controlled potassium ion gates. For the Smarties, the yellow Smarties are potassium ions, the pink are sodium ions, the green represent proteins, and the brown are chlorine ions, and the red are ATP. And orange Smarties are synaptic vessels, and purple Smarties are neurotransmitters. Establishing the resting potential of any one part of the neuron is done through the use of an ATP-powered sodium-potassium pump that moves ions against the gradients, which pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. This establishes a potential difference with outside the cell being positive with its stockpiled sodium and the inside of the neuron being negative with its stockpiled potassium ions. This is at about negative 70 millivolts. Proteins and chlorine ions are also present inside the neuron, assisting in establishing that negative charge. This resting potential difference is the starting point of every nerve impulse and must be in place for an action potential to occur. Excitation is when the dendrites of the nerve cell experience a stimulus such as heat or pain, and many others. This stimulus causes the voltage-controlled sodium gates to open a bit and allow just a little sodium to leak into the cell. Now, if the stimulus was strong enough and enough sodium ions got into the cell, changing the potential in the cell from negative 70 millivolts to negative 55 millivolts, then the nerve cell will fire. This response is an all or nothing response, so if the stimulus isn't strong enough and doesn't allow enough sodium ions in, the resting potential isn't changed enough and the neuron doesn't fire. This value is called the threshold potential, and it is negative 55 millivolts. Any change in potential less than this and the nerve cell will not fire. Any change above the threshold will cause the neuron to fire to its maximum voltage each time. A note to make is that when the neuron fires, it fires at the same strength each time, disregarding the strength of the stimulus. Instead, when the cell encounters a stronger stimulus, a neuron will fire more often or more nerve cells will fire. Depolarization is the loss of a negative charge in a nerve cell. This occurs when a threshold potential is met and an action potential begins. When the neuron hits the threshold potential, the voltage-controlled sodium gates open, allowing sodium ions to travel across the membrane down their concentration gradient. This creates a positive charge on the inside of the cell and a negative charge on the outside. The change of potential is the same each time. It is from about negative 70 millivolts to about positive 40 millivolts. This rushing of sodium ions into the neuron is what makes up a nerve impulse. Repolarization is the storing of a negative charge in a nerve cell. This happens once all the sodium ions rushed into the cell and we hit about positive 40 millivolts. This causes the potassium voltage-controlled gates to open, allowing potassium ions to rush out of the cell, restoring that negative charge on the inside of the neuron and the positive outside of it. Hyperpolarization is when the inside of the cell is too negative. This occurs because the potassium voltage-controlled gates don't close right away, allowing more potassium ions to leave the cell as the potential overshoots to about negative 90 millivolts, instead of the needed negative 70 millivolts. This needs to be fixed. A note to make is that the neuron can be hyperpolarized at rest due to inhibitory neurotransmitters, making it harder for the neuron to fire. A nerve impulse is the rushing of sodium ions into the cell, so after a nerve impulse, we see potassium ions outside the cell and sodium ions inside the cell. This is wrong, and to get the neuron back to rest and ready to fire again, we must move the sodium out of the cell and the potassium back into the cell. This is what occurs during the refractory period. Move the sodium out and the potassium back in. Without this, the nerve cell would never be able to fire again. This process is done through the same device that maintains a cell resting membrane potential, the sodium-potassium pump. But now it is just working extra hard to move everything back to where it needs to be. It uses ATP to move 3 sodium ions out of the cell and 2 potassium ions back into the nerve cell. Signal propagation is the movement of the nerve impulse throughout the entire nerve cell. It is done by the diffusion of sodium ions through the cell. A nerve impulse is sodium rushing into the cell, and when it occurs, the sodium that rushed in diffuses to the next portion of the neuron. This increases the potential to the threshold value, and another action potential occurs at that portion of the cell, and more sodium rushes into the neuron. This process continues all the way to the synaptic bulbs. The reason it is able to move in one direction is due to the fact that for the nerve impulse to travel, it is done by diffusion of sodium ions, and the sodium will not diffuse backwards since there's still a high concentration of sodium ions in that portion of the nerve cell, as it just underwent an action potential. This causes it to always move forward through the length of the nerve cell. In an unmyelinated neuron, the sodium ions have to diffuse and open every sodium voltage-controlled gate along the length of the axon. For a myelinated neuron, the Schwann cells are wrapped around the axon, preventing the voltage-controlled gates from opening. However, there are places between each Schwann cell called nodes of Ranvier where sodium voltage-controlled gates are able to open, and the sodium ions are able to diffuse and skip past to each node of Ranvier. This skipping past portions of Schwann cells and jumping from node to node in myelinated nerve cells allows for much quicker transmission of nerve impulses compared to unmyelinated neurons. Synaptic transmission is the process of transmitting nerve impulses between nerve cells and or transmission of an impulse between a neuron and an effector. This is done through the use of neurotransmitters. As the nerve impulse reaches the synaptic bulbs, the synaptic vessels bind to the membrane of the presynaptic neuron. This releases the neurotransmitters and they diffuse across the synaptic cleft to the postsynaptic neuron. The neurotransmitters bind to postsynaptic receptors and cause either an excitatory or inhibitory response. If excitatory, then sodium-gated channels open, allowing an action potential to begin. However, if inhibitory, then potassium channels open, making it harder to fire that neuron. After the neurotransmitter causes its intended response, it is broken down, and we see this in the example of acetylcholine and cholinesterase. Acetylcholine causes its often excitatory response, and afterward, cholinesterase quickly breaks down acetylcholine so the neuron stops firing.